As I said, it's a privilege to be here. I'm the only academic speaker on the agenda here. No, I mean industrial speaker. I'm the only industrial speaker on the agenda. And it's funny, I received a note from Eddie Rubin about a week ago. I wasn't sure if this was associated with the fact that I was the only industrial speaker on the agenda, but it said, first of all, there's going to be a diverse range of people here, which today's talks absolutely, he was spot on with that comment. The second one was, don't be afraid, and in fact, don't avoid presenting cool science, right? Cool science, because I know some industrial talks can be, shy of the science. So I'm going to try to present some cool science associated with what we're doing. I'll let you know when it's coming, just so you know. But overall, Genomatica is focused on not curing cancer. That's not what we're doing, but we're trying to solve, or at least participate in a small way. We're a small company. Participate in addressing another major issue, and that's climate change. So I'll give you a little introduction. We're a technology company focused on the production of sustainable chemicals. We're a technology provider to the chemical industry. So when you think about chemicals, chemicals are actually good. Chemicals are actually good. In fact, you can't really look around you anywhere you go in society without seeing the impact of the chemical industry. Chemical industry really shapes our lives. Many of the things that are related to our standard of living today, especially in the developed world, come from the chemical industry. And these are just a few examples that I'm showing here. And I don't know if you recognize the guy I just flipped up there at the top there. That's actually Clint Eastwood. So that gives you a little preface in terms of where this is going. So this is the good. The good is the chemical industry does supply us with useful materials, useful products. And I'll give you a little more insight into that in a second. Now we get to the bad and the ugly. The chemical industry actually has a bad reputation, and chemicals in general have a bad reputation. The bad, that's Levon Cliff. He was a nasty guy in this movie, The Good, the Bad, and the Ugly. You see the bad, the explosions, the pollution, the toxic waste that's generated by the chemical industry. There's also the ugly being, the refining infrastructure that we have. I'm sure a lot of us have been to Santa Barbara. It just kills me every time I go to Santa Barbara, looking off that beautiful coast and seeing those oil rigs sitting out there. It just kind of destroys the view. And then that bottom picture on the right is the Pacific garbage patch. That's that big, basically Texas-sized collection of plastics that all the currents have collected in the middle of the Pacific Ocean. That's some guy in a canoe actually wading through that plastic patch, all coming from chemicals, right? And so, if that's not enough, CO2 is becoming an increasing issue here. We're seeing rising levels of CO2. 40 billion pounds of CO2 have been emitted this past year. 40 billion pounds. Atmospheric CO2 is now, February of 2015, this is data from 2014, but February of 2015, we were over 400 ppm CO2 in the atmosphere. And this is all linked to climate change. This is all linked to global temperatures, highest year, 2014. Land and sea global temperatures average over the year the highest ever recorded in 135 years of recording this data. So you have half the global CO2 emissions from the US and China. That's 20 billion pounds of the 40 billion pounds is coming from two countries. You have high CO2, certainly linked to the rising global temperatures. You have glaciers. I just saw some, I don't know if you've ever seen this show called Vice, which is an investigative reporting show, which is really quite a good one, that they've gone to Greenland and they've gone to Antarctica recently and shown some pretty massive melting that's going on in those two areas. So the glaciers are melting, the oceans are becoming more acidic, and this is all due to this massive infrastructure that we have that supports the standard of living. And so the question is, what can we do about this? What can we do about this? If you look at the current industry, it's based on fossil fuels. So you go from oil, gas, coal, especially in China, going down to basic chemicals, raw plastics, and then finally to the products. I'll give you a little more detail. How chemicals are made right now. You look at natural gas at the top, or oil going into naphtha. It all goes into a cracker, and that cracker, right in the middle there, that steam cracker, leads to the basic 7 chemicals that are used to make everything else that the chemical industry makes, essentially. So you have those 7 chemicals coming out of crackers. So if you hear of crackers, it's either taking gas or oil, cracking it into those materials. On the right are pictures of crackers. Beautiful, aren't they? Nonetheless, they supply what is needed to produce the products that we have of interest. So you go from a basic chemical, intermediate chemical, materials, and then the brand products. Here we have ethylene, xylene, acetylene, propylene. And here's the whole product chain. Here's the value chain in the chemical industry. You have ethylene going to ethylene glycol and polyethylene. Polyethylene is the bags on the right-hand side. Ethylene glycol combines with terephthalic acid to give you PET. That's all our plastic bottles that we're drinking out of. You have PBT, which is also a material used in the automotive industry, but that's combined with terephthalic acid and 1,4-butane-diol. 1,4-butane-diol is also the main component of spandex. And probably most of us, somewhere on our body, are wearing spandex. Well, that's coming from oil right now. Also propylene, polypropylene. We have polypropylene everywhere, along with polyethylene. And finally, propylene also goes to acrylic acid, which goes into superabsorbent polymers, which we put on our babies. So all this is coming out of the chemical industry. And as it turns out, all of these products and almost everything you see around you that isn't either living or once living, wood, for example, mineral or metal, comes from oil, coal, natural gas. And so what's Genomatica focused on then? Okay, so Genomatica is trying to help address this. We're a small company, as I said. We can only do a little, 1 chemical at a time, for example. But we're trying to play a role in addressing some of the challenges that I just noted that the chemical industry is facing and that we as a society are facing. So that's associated with using renewable feedstocks for the production of these chemicals. So Genomatica is focused on sustainable chemicals, and how we do that is the production of high-performance organisms and processes, and engineering those organisms to produce the basic and intermediate chemicals that the chemical industry needs, and downstream applications are required to produce the products that we want. This is what we want. And how we do that is by identifying and engineering pathways, and quite often the chemicals that we're producing are not naturally produced by any microorganism. So we have to actually engineer novel pathways for the production of these chemicals, introduce those into a microbial strain. I can tell you that microbial strains are not happy when you do that. So there's a lot of engineering that has to go on to get that organism to be comfortable, and not only comfortable, but robust, by allowing you to funnel its carbon energy redox down the pathway of interest to produce as much of that chemical as it possibly can. Okay? And it still has to be robust at commercial scales. That's part of the challenge. So what we've done is we've developed a technology platform and a workflow that's shown here. And this workflow really centers on a design, build, test, learn cycle around which we wrap technologies. And the technologies start with the metabolic modeling and model-driven design and data analysis, all the pathway engineering, strain engineering, a rational approach to strain engineering involving systems biology. I'll be telling you about that. Automation, small-scale screening, and reproducibility. We talked about it. It's so important because as soon as you get up to statistical analysis, if you don't have reproducible data, you might as well throw out that analysis. You need reproducibility in your assays, and the data has to be consistent. Fermentation, we rely heavily on omics analysis, very deep omics analysis on a weekly basis, as I'll show you, followed by a LIM system for data management and statistical learning for really knowledge creation. This is our workflow, and we use this and leverage this for the engineering of microorganisms. I'll show you how we engineered an organism for the production of 1,4-butane-diol. This is the chemical in our portfolio that's most advanced. And it's a chemical that goes, 1.7 million tons are produced each year. It goes into the production of PBT, TPUs, and spandex. I mentioned spandex before. The main component of spandex is 1,4-butanediol. So again, we're trying to do our little part to help the industry become more sustainable. So this is a direct production from sugar to 1,4-butanediol, which is sold commercially. And we've used detailed systems and diagnostics experiments that I'll show you in order to develop this process. So if you look at the pathway, we engineered E. coli, take up sugar, go through glycolysis, TCA cycle, a BDO pathway, producing, right now we're producing about 140 gram per liter of 1,4-butanediol. It's a lot of 1,4-butanediol, 14% solution of BDO is produced with this microorganism. That's the stoichiometric equation for the production. We get about 0.5 gram per gram, 67 carbon mole percent yield as a maximum yield, and we're pushing that yield up higher and higher all the time, and I'll show you how we did that. Here's the pathway. You can see the pathway. The last 5 steps of this pathway coming off of succinyl-CoA, off the TCA cycle, are not naturally occurring. So each of those enzymes is working on a non-native substrate. And that's important because when it's working on a non-native substrate, it's probably not optimally active. And quite often you encounter inhibition. And so you need engineering techniques, protein engineering techniques, in order to overcome those challenges with activity and inhibition with those enzymes. This is a redox-intensive pathway. And as you can see, the pathway that we've engineered inside the microorganism, we push the flux down the oxidative branch of the TCA cycle, and that's in order to generate enough redox to power that pathway. In addition, we have excess redox that we're oxidizing with the oxidative phosphorylation pathway, and that's required because through this pathway, it's an ATP neutral pathway, so there's no extra ATP that's generated in order to facilitate growth and maintenance of the organism. And so that redox is really needed in order to allow the organism to survive in a robust manner. So if you look at the pathway going from glucose all the way down to BDO, as I mentioned, we're going down the oxidative branch of the TCA cycle. The organism quite often has other ideas. We're trying to produce 1,4-butanediol. The organism has other ideas. Right? Here are all the byproducts that we have encountered in our desire to engineer the organism to produce 1,4-butanediol. These are all the different byproducts that we've encountered. And in general, the message that I have is the byproduct formation occurs for a reason. Right? You're trying to engineer E. coli to produce 1,4-butanediol or some other chemical. The organism wants to grow. Right? So it's going to want to supply itself with energy and redox, and balance its redox in a certain way, and it's going to do whatever it can, and quite often that entails production of other byproducts. And so it occurs for a reason, and it's usually satisfying some metabolic or physiological need that it has. And so what we've introduced is a lot of systems analysis in order to really decipher the reasons and the causes of those byproducts that are being formed so that we can get to the root cause of why that's happening so that we can circumvent those byproducts from being formed, because those byproducts are a reduction in yield for us. And a reduction in yield is not bad. So today what we're going to focus on is 4-HB, which is down at the bottom. 4-HB is down here at the bottom. That's a byproduct that we're seeing significant amounts of. And then CO2 loss. CO2 loss occurs across numerous places within the cycle. And so I'm going to show you, here's where we're going to get into some of the cool science, I hope. I'm going to show you how we address these challenges in the production. So what we've done is, what I'm showing you here is improvements that we've made to the strain. And this is tighter over time. Over 4 years, we've been able to really ramp up the amount of BDO we're producing and the rate at which we're producing BDO to a level that's significant with regard to commercial production. So it's all about getting the economics to be appropriate so that we can compete with those petrochemical processes that I told you about at the beginning of the presentation. To get the economics, in fact, you need the economics before you can ever commercialize. No chemical company in the right mind will commercialize a competitively uneconomic process. And so you need to develop the process for economics, and this requires you getting those tighter rate and yield as high as possible. What we've done is we've implemented an iterative multi-omics approach which led to these advances. And we use all the standard technologies, and we do this on a weekly basis. As we're engineering the strain, we need to know what's going on inside that strain because, again, it's reacting. It's responding to all the changes we're making. We need to know what those changes are in order to draw new hypotheses and to go to the next step. And we also introduced those network analysis tools as well as machine learning framework in order to analyze the data. And designing good experiments is really crucial here. So the first thing we looked at was 4-HB. 4-HB spikes. It spikes when the BDO hits high levels. Here we see BDO leveling off, 4-HB right at that time starts popping up. That spike occurs, and so all of a sudden you're producing a lot of 4-HB when you don't want to. And so the question is, as that's happening and you're producing all this 4-HB, why is that happening? Why is BDO leveling off and why is 4-HB spiking? How can you decrease that 4-HB? So we did a pretty cool C13 flux isotopic tracing experiment where we've taken glucose. If you look at glucose coming through down the BDO pathway, there are 3 main enzymes leading to BDO formation. 2 of those enzymes, ALD and ADH, also lead to ethanol production. And so we need to distinguish between those 2 pathways. And what we did was we looked at the isotopomer ratio changes that occurred upon feeding C13 glucose, non-labeled 4-HB, C13 BDO, looking at the unlabeled BDO that's formed from 4-HB going down this pathway. In addition, we've also added non-labeled ethanol and looked at C13 labeled ethanol coming from C13 glucose. And the upshot of that, the results of that experiment were taken over 2 different time points. And what you can see is the first time point, what we see is at high concentrations of BDO, what we observe is a decrease in BDO production at higher concentrations, but no decrease in ethanol production. No decrease in ethanol production, meaning the ALD is not impacted. ALD is not impacted in this experiment, but CAT2, this enzyme right here, is impacted by high levels of BDO. So CAT2 is inhibited by high levels of BDO. Then at the later time point, what we see, in both cases, we see a decrease in activity of the formation of BDO. And so what that's indicating to us is at the later time point, ALD activity is dropping. So what we did to address this is first focus on CAT2, and this is what we've done for a range of different enzymes now to improve the activity of CAT2. What we've done is implemented a discovery. That's we take all the known CAT2 enzymes, create a pipeline, and screen them, and then we find the best one under high concentrations of BDO that's active, and then we evolve it for higher activity. We evolve it using the standard methods of mutagenesis and high throughput screening to come up with a better enzyme. And here we found an enzyme that's 20 times more active at 1 molar BDO. So that addressed the CAT2 issue that we were facing. Similarly, for ALD, you can see on the right-hand side of this Western, you see the ALD decreasing over time. Well, we did the same thing, implemented the same strategy, discovery followed by evolution, found an enzyme that's 10 times active and lasts a lot longer. And so over the course of the fermentation, you see no change in the level of ALD. Interesting phenomenon, however, is with the parent enzymes, here's the level of activity. We put in the evolved CAT2, you actually get lower amounts of BDO. You put in the evolved ALD, lower amounts of BDO. You had to have both of them in there in order for the desired effect to take place. 15% increase only when you have both evolved enzymes put in the organism. And so the message there is, and I'll have one more message along these lines, multiple strain changes are often needed for improved activities and improved performance of these engineered strains. So overall, what we're seeing is 4-HB, much less being produced and at a much later time relative to previous studies without the evolved enzymes, but yield is still too low. And what we're seeing is biomass and excess CO2. Biomass and excess CO2 are still predominant in terms of yield drains from the system. So what we wanted to do, and you can see how 4-HB has gone down significantly following those previous studies. So the next thing we did was looked at oxygen levels, and oxygen is really important in these systems. reducing oxygen supplies does lead to an increase in yield, but it also leads to a reduction in BDO. So here you see that, CO2 going down under low oxygen, yield going up under low oxygen, all that's really good, but you see BDO rate and growth going down under low oxygen. And so what we needed was diagnostic omics engineering required to increase both the rate and the yield under low CO2 conditions in order to get yield up. We need yield up. 3 approaches: reducing flux through the CO2 forming pathways, increasing efficiency of ATP production, and finally NADPH levels. You had to address all 3 of these at the same time. So what we see is reducing flux through the CO2 forming pathways. First thing, we saw the three CO2 forming pathways. Here, here, and here. This is all the TCA cycle. We reduce the pentose phosphate pathway, that's a WIF deletion, reduce oxidative TCA, and finally the glyoxylate shunt. And what we found is actually that BDO decreased, and the excess CO2 actually increased under those conditions. And so we did a pretty detailed systems analysis of the system to understand that. It didn't make sense. We're knocking all these CO2 forming pathways out, and all of a sudden CO2 goes up. Well, what was happening there? So we did a systems analysis, and we found that the E. coli actually had an enzyme that served to complement that SUCCD deletion, and it's what we call a rogue flux. That rogue flux led to completing the TCA cycle, and as carbon keeps going around the TCA cycle, the organism gets redox, it gets ATP, and it leads to CO2 loss. So we had a complementing factor inside the organism. We then, and so that excess CO2 pathway is actually leading to that something that the organism needs, Deleting that rogue flux also led to very poor growth of the organisms. So overall, what this is telling us is that you need more ATP, we need more NADPH. ATP, we looked at deleting potential ATP drains, ATP dissipation cycles such as PEP-CK, PPC cycle, deleting unnecessary cellular functions such as the flagella and the fimbriae. And also, we looked at the oxidative phosphorylation. We looked at the electron transport chain. And what we found is that there are several very inefficient parts of the electron transport chain, some more efficient, NUO, CYD, CYO, very efficient. So what we did was we knocked out the inefficient elements of the electron transport chain, leaving only NUO and CYO to generate the proton motive force. And lo and behold, we did see an increase in growth, as expected, or as hoped for, I should say, and also a slight increase in BDO, but not where we needed it to be. And that gets us to the last part, which is NADH, NADPH. C13 tracer studies indicated a high flux through the pentose phosphate pathway, and that indicated a need that the organism has for NADPH. That need also suggested that if we have excess NADH, there should be a way of converting that over into NADPH, and in fact, E. coli has a transhydrogenase that allows you to do that. Overexpressing PNTA-B, which is a transhydrogenase, enabled us to convert NADH to NADPH, and that led to limited pentose phosphate flux as well as lower CO2. And so combining these, the CYD deletion, which leads to higher ATP, plus the PNTA-B overexpression for NADPH, led to significant increases in BDO, significant increases in growth, as well as decreases in excess CO2 and then increases in yield. So once again, what we're seeing here is combined changes are required when you're engineering microorganisms. Combined changes are required for getting the improved performance that you're looking for. Finally, the third challenge, a very short story. So here we have genetic instability, and that's really a killer when it comes to commercial production. If you have a genetic instability in your strain, trying to produce a commercial scale, what you're going to see is declining performance over the course of the fermentation. And what we had observed, this is quite recently as it turns out, and we weren't very happy about that. A new batch of bullets that we tested for BDO production had performed very poorly. We didn't understand why, so we did whole genome sequencing using MiSea. We have in-house MiSea, revealed a large duplication event that was occurring. And here's that duplication event. You can see right in the reads right there. And so that duplication event was actually a fairly significant region, a 321 kb region. with a range of different functions, including essential genes. So you're duplicating that. Some proteases leading to key pathway enzymes being degraded. And so that was a big problem that we were having, and that's why the BDO production went south. And so what we ended up doing was as it turns out, the region borders here actually had 2 cryptic prophage and 2 IS elements. We looked at the prophage. That didn't do too much. In fact, it was detrimental. But finally, deleting those IS elements, we found that flanking IS elements actually addressed the problem. But we wouldn't have found this nearly as fast as we did and wouldn't be able to address that kind of a challenge without having that MiSeq instrument in-house. And so that really greatly facilitated our ability to solve this challenge that we're facing. So putting it all together, this is what it takes. A kind of a take-home message today is if you're engineering microorganisms for specific functions, you need multiple changes quite often and have to put it all together in the same strain in order to observe the big increase in your performance. And so you saw the increase that we observed over 4 years. It took us 4 years. That sounds like a long time. But over 18 months, using this technology platform that I showed you earlier, you can see the significant improvement in BDO production and a reduction in byproducts that we're seeing. Combined changes, as well as increasing the yield and a genetically stable host resulted from this, ultimately leading to the commercial targets being exceeded. If you think about commercialization, here we're commercializing BDO. Over the course of 5 years, we went from production at the left-hand side, 5 milligram per liter, up to 140 milligram per liter, commercial scale production at 600,000 liters scale. We commercialized this process and operated it at actually a plant that was built to produce 1,3-propane-diol. We were producing 1,4-butane-diol in a DuPont Tate & Lyle facility in Loudon, Tennessee. Technology platforms really aimed at driving timelines, accelerating timelines, and reducing costs. This technology platform I told you about, we've used it for another product, which is 1,3-butane-diol. Ultimately, that's a precursor to butadiene. Butadiene is used for things like synthetic rubbers for tires. So a lot of tires are produced from butadiene using synthetic rubber. And so butadiene is a very, very large volume product globally. One of the challenges with the U.S. and shale gas, you might have heard of shale gas in the U.S., shale gas is being used now as a lighter cracker feed. In fact, the ethane coming out of shale gas, lighter cracker feed, that's leading to less butadiene produced in the U.S. because of the lighter cracker feeds. So butadiene is an important issue. And what we found is over 10 months, I just told you about 4 years, over 10 months, we were able to produce significant levels of 1,3-BDO. In fact, if you compare it to 1,4-BDO, it took us 27 months to get to 51 gram per liter. It took us 10 months to get to 54 gram per liter with 1,3-BDO. And that's all about the technology platform. This is about leveraging that technology platform I told you about, and also the experience in how to use these kinds of technologies. It's really important to have that integrated technology, tightly integrated technology platform. And finally, I'll just end by saying we're looking for other opportunities to leverage this technology platform. We're going from the integrated technology platform to engineering microorganisms and looking at precursors and cofactors and optimizing flux, balancing ATP redox, eliminating byproducts. We've done this for a range of commodity chemicals. We're also now expanding that into more complex molecules such as natural products. This is an area that we think is really exciting, and we're driving toward that, leveraging this technology platform. So with that, I will end and just thank the people that have been involved at Genomatica to really make this happen. And so I thank them, and it's all about the people, and I love them all. And finally, I'll thank you for your attention. This is our building in San Diego. Thank you, and I'll be happy to answer any questions.
Questions before the break? I was curious what prevents loss-of-function mutations in the pathway leading to competition of your production strain in batch?
Yeah, well, the nice thing about batch is it's only for a short period of time. And the number of doublings you have to do at commercial scale, the number of doublings is fairly small. It's only about 30 to 35 doublings that you're engaging in. So on average, you don't accumulate the mutations. Plus, you're integrating these. You're not overexpressing these on a plasmid. You're integrating them into a chromosome, single copy, and so they're nice and stable. And so you're not seeing that many mutations. In fact, we do whole genome sequencing all the time, and we're surprised at how little we see mutations occurring in the heterologous genes.
Okay, question?
So make plastic from sugar, cost-wise, how does it compare with cracking oil? Is it competitive or?
This process that I showed you, 1,4-butane-diol,
Yeah.
there are two processes that are currently available that people can build new facilities with. We are actually cost-advantaged relative to the incumbent petrochemical processes. So companies such as BASF is the biggest 1,4-butane-diol producer on the planet, right? And they have licensed our technology for the production of 1,4-butane-diol.
Okay, why don't we take a break and start back right up